Hello, everyone. Thank you so much for attending the open house today. We're so excited to tell you about the various things that we do here. So I help manage Tissue Portal, which is a group that performs high quality biospecimen processing for translational research. Can everyone hear me online? All good? Okay. All right, so biospecimens are precious. I don't need to tell anyone in this group how difficult it is and how resource intensive it is to collect samples and how much trust patients and donors need to have in us to provide samples to us for use in cancer research. As such, it is our responsibility to make the most of these biospecimens, ensure that they are used in an ethical manner for studies today, as well as preserved for future studies. Tissue Portal is really committed to this, and we provide high-quality biospecimen processing for a variety of different cancer research studies. We ensure that specimens received for fee-for-service requests have all the ethics documentations, so we will ask you for your Research Ethics Board approval anytime that you submit samples to us. We act as a centralized tissue processing and histology core here at OICR and support many of the different teams within the Institute, as well as external researchers. We need to meet all of these tissue processing needs in a cost recovery way. We are a nonprofit here at OICR, so we must use a nonprofit model for our pricing. I'm going to take you through the sample life cycle of a sample as it comes through Tissue Portal and give you examples of our various services that we offer, as well as our connections to other teams at OICR. It all starts with the donors that will be part of clinical trials and donate their specimens for cancer research. The external lab where these samples are collected, or the hospital, will have a formalin-fixed paraffin-embedded tissue. This is symbolized by the blue block up at the top. Fresh frozen tissue in the green capped vial there, and blood that is collected from the patient. Blood is then processed and fractionated into plasma and buffy coat, and these different sample types are the ones we most commonly receive at Tissue Portal. These are sent to us, and then we perform a number of different services as needed for the project. We perform basic histology by sectioning FFPE, and we create unstained slides as well as stained slides. We do stain with hematoxanil and eosin for pathology review, and we can also create tissue microarrays where we take cores from multiple different donor blocks, put them all into one recipient block, and then assays can be done on multiple cases, 200 cases at once on those slides, instead of having to process the whole slide sections. We also digitize H&Es and TMAs. So we have a slide scanner that we use. This is how we create the virtual library that exists for the Ontario Tumor Bank, as well as for other studies such as Octane. For fresh frozen material, likewise, we also do sectioning. We do a lot of cryosectioning and staining. And then we can use this material for diagnostic development assays. So we pass these samples off to the diagnostic development translational research team, where they can perform digital spatial profiling, which you just heard about, or IHC. Samples are also enriched. Often we enrich for tumor cells, tumor cellularity, because that's what most of our studies need. But we've also worked on enriching for stromal compartments, as well as different histologies within a tissue. We do this either through macrodissection, which is manually done using a scalpel to scrape off areas of interest, or using a laser capture microdissection system, which I'll talk about a little bit more in detail later. The bulk of our work is performing nucleic acid extraction. We do a lot of this. So we will take fresh frozen tissue and either section it or cut it into smaller pieces and homogenize. And then we take enriched tissue, lysed tissue, plasma for cell-free DNA, and buffy coat, various blood components to perform extractions where we get DNA and RNA. These samples are then taken through quantification and qualification. We use fluorescence as well as absorbance to quantify and qualify our material to verify the purity and the quantity of material. There's a picture up here of barcoded tubes reminding me to tell you that our entire process is barcode tracked. So we have a very robust LIMS system in which we track the progress of samples throughout our entire life cycle. And this helps us keep on track with the large number of samples that we process on an annual basis. DNA and RNA can then be transferred to the diagnostic development team, where they can perform nanostring assays, as well as using the S5s or other Thermo Fisher sequencing systems for panel-based assays. And the material is also transferred to our genomics team that will talk to you later about the plethora of different sequencing solutions that they offer. All of these different sample types are also stored with us on a short-term basis. So we do offer short-term biobanking for investigator-led studies. We store both at liquid nitrogen and at -80. And then once a study is completed, we will return these samples, or we can also distribute the samples to third parties as requested by the research team. The researcher receives the rest of their samples back. This includes the original sample that was provided to us, if there's any remaining, along with any nucleic acids that are remaining. What this does is it helps ensure that these samples can be used for future studies. This is especially important because biospecimens are precious. Data is also returned from the diagnostic development and genomics teams to researchers. And one other thing that I wanted to mention is that we really do have a commitment to quality in our group. We have various QC gates that samples go through. We verify all of our processes with controls, and we also keep track of all of our equipment, performing the necessary preventative maintenance as well as calibrations on a routine basis. We have two different but very similar assay streams at OICR. We have the research use only stream, which we use primarily for academic and industry research projects that might be a little bit more experimental. A clinical report for a patient is not needed. Perhaps it is something slightly unusual that needs a boutique service. So we do histology and nucleic acid extraction for this. We also help with correlative sciences biobanking, as I mentioned, and we also have collaborative opportunities for technology development. So if there is a device or assay or something that you want to test or validate, and our team can help with that, we're happy to discuss and see how we can chart a way forward. Our validated clinical assays, or as I call them, VACA assays, are part of the OICR Genomics Laboratory, which includes Tissue Portal and the Translational Genomics Laboratory. We provide nucleic extraction services for next generation sequencing. And to anyone that is wondering what a validated clinical assay is, it's an assay that has been proven through objective evidence to perform reliable diagnostic testing with both precision and accuracy. We're able to do this because we have our triple accreditations, and more information about our quality management system and how we're able to do this will follow in presentations later today. As a case study, I wanted to talk to you about the PanCure group. This is a collection of studies that are performed as part of this translational research initiative, which is headed by Dr. Julie Wilson, who is the Associate Director, as well as the PIs, Dr. Stephen Gallinger and Dr. Jennifer Knox. We've been working on the various studies under this umbrella since 2012. And it exemplifies a good range of our services, as well as how we're able to utilize both of our streams for making an impact in cancer research. So it all starts with the donor. They will be consented to a research study, and biopsies or other SNAP frozen tumor tissues come to Tissue Portal. We'll perform cryosectioning, and an H&E is created. This is hematoxylin and eosin stain section, which is reviewed by a pathologist and marked up for regions of tumor. We also section an additional set of slides, which is stained with crescent violet. Crescent violet is a stain that helps us visualize the morphology while also not detracting from the nucleic acid quality or quantity following extraction. For laser capture microdissection, this is the next step, and this is where we take the violet stained slides and we select portions of them to be able to isolate them for extraction. This is particularly important in pancreatic cancer, where you can have a very low tumor cellularity. The average sample can be about 20% tumor and 80% stromal or normal cells. And you can imagine if you were to sequence this in its entirety, that normal signal will drown out the tumor signals. So what we do is shown here, and I cannot tell my left from my right, but the triangular piece of tissue stained in pink at the bottom there is a piece of tissue from an excision. And the little rectangle that is pictured there is our field of view while we're performing LCM. It's a very laborious technique. You can see that the larger pink image with the green outlines, this is the H&E, where a pathologist has shown us where these tumor cells are located. And then a skilled operator is going to real-time mark out these same areas or similar areas on the crescent violet stain section, which is the blue one a bit closer to me. All of these different areas are then captured in a tube, and we can perform nucleic acid extraction to get the tumor DNA and tumor RNA. And frozen buffy coat is used to get germline DNA that is used as a reference for sequencing. Nucleic acid extraction happens, and these nucleic acids are passed to TGL, where they do their sequencing magic, and then GSI creates a clinical report that is passed on to the medically responsible physician for that patient, and they can use the information contained therein to help guide the patient's treatment. So this is a direct impact to patients right now in real time. There is also research data that is generated, and all of this is compiled over time and analyzed by very skilled and knowledgeable individuals, and we help contribute to the overall body of knowledge that exists. These are just some of our publications from the PanCure team, which have helped create more understanding about this disease for future patients. For correlative sciences consultation, this is a bit on the side of everything else that we talked about, but because we really have an understanding of what is needed from collections of biospecimens to be able to use those samples for next generation sequencing and other downstream assays, we offer our expertise to be able to assist others in putting together lab manuals, associated forms, and sample kit collections. For example, we assist with the WU trials as well as the PRECISE trial, which is ongoing, and we're always happy to lend our expertise to anyone that might have some questions about how to put something like this together. We work with many different groups. As you can see here, these are just some of the ones mostly centered in Toronto. We're part of the Marathon of Hope Cancer Centers Network and help with the Biospecimen Working Group that helps to guide the network's use of biospecimens for the initiative. We work with research laboratories, hospitals, and industry clients across Ontario, in Canada, and also globally. And our pricing, an example of our most commonly used services, is shown here. Again, this is a cost recovery model. It is nonprofit, and depending on the scope of your study and additional services that you may need, we may be able to come up with a pricing structure for you that has a discount if we're able to do these assays in a high throughput manner. That goes after this one. So overall in Tissue Portal, excuse me, looking at the wrong image here. So overall, our volumes are quite high. We receive these 4 different images here. They have different colors. The lighter colors in each of these graphs actually shows the COVID viral work that we did during the pandemic. So cancer research kind of ground to a halt. And so we shifted our focus at the time, and we were one of the first teams back at OICR trying to work up extraction protocols to figure out how to extract COVID and then be able to help with contact tracing in the hospitals for this. So that accounts for some of our volume kind of strangeness that is shown here. On average, we receive about 27,000 samples per year. And you can see in the current fiscal year, we're only at 6 months and we're set to exceed that amount. Usually we prepare about 1,800 samples for sequencing. This year, we've already exceeded 5,000 samples sent to both our genomic sequencing team here, as well as external centers across Canada. We perform about 3,500 extractions per year, but we're set to exceed that number this year as well. And our distributions, which are either releases to third party on behalf of researchers or distributions from our internal investigator-led biobanks, these are not sample returns to researchers, are about 3,000 per year, and those have remained relatively stable. But how do we cope with these increasing volumes of samples that are coming our way? And in Tissue Portal, we've taken an active role in trying to improve all of our processes and our methodologies. So a few of the things that we've done is that we're currently working on bringing blood fractionation in-house. This helps us control the quality of plasma that we get for cell-free DNA extractions. So that is work that is currently ongoing. And for nucleic acid extraction, we've really adopted high throughput methods. So semi-automated systems like the Kingfisher, a magnetic bead-based extraction system, and liquid handlers help us work on a plate basis. So instead of working with 24 samples at a time in a batch, we're now able to do 88. It's a 96-well plate, but we need room for controls. So 88. As well, our quantification and qualification is also being improved over time. So instead of relying on qubit, which is one sample at a time that you check your quantities of, we're now using plate-based readings instead. About 75% of all of our protocols are now worked up on these high throughput systems, and this allows us to be able to cope with the ever-increasing number of samples that come our way. How do you access our services? This giant QR code here, it will bring up your mail app. Press send, it will send me an email and I'll set up a meeting with you. You can also contact me at that email or email Diagnostic Development or Tissue Portal directly. Project setup will include a consultation at first. For simple projects, this can be done through email. Otherwise, we will do this through setting up a meeting with you. A project information form will be asked. I know no one likes to fill out forms, but it really helps us capture all the information needed so we can process your samples in the way you need them processed. And then there's an estimate approval that needs to occur. Your REB approval letter from your external REB needs to be submitted to us before samples come our way. And then a sample submission form with all the necessary information about how your samples are labeled and what we should be doing with them is submitted at the time of sample receipt. If you are obtaining samples from the Ontario Tumor Bank for your study or you're performing assays with OICR Genomics, the process above is really tied into the entire project life cycle. So you don't need to submit two separate sets of forms for these different groups. This is our team here. So our co-directors who you just heard from empower us to do the large amount of work we do. And our biospecimen research technicians are really conscientious and hardworking. And they help us meet our mission in Tissue Portal, which is to help you as researchers get the most out of your biospecimens.